Rift Valley fever virus. Young sheep are dying on a farm. Abortions are also evident in cattle. Rift Valley fever virus is suspected. Which possible samples would you take to send to the laboratory? The first step to take when trying to identify the Rift Valley fever virus is to collect some samples. These samples can be heparinized or clotted blood, blood plasma or serum, and tissue samples of the liver, kidney, spleen, lymph node, blood of the heart and brain from dead animals or aborted fetuses. Then these samples should be preserved and transported to the laboratory for further testing. Specimens should be submitted preserved in 10% buffered formalin and in glycerol or saline and transported at 4 degrees Celsius. The samples or specimens must be transported in 3 layers. The first layer contains the sample, the second is a protective layer, and the third is a solid and rigid layer. Liver or other tissue for histological examination may be placed in formal saline in the field for diagnostic purposes, which facilitates handling and transport in remote areas.
Will you take extra precautions sending these samples to the laboratory? If so, why and which precaution? We have to take extra precautions sending these samples as we might be dealing with a virus. Samples should be kept cold during transport as this reduces bacterial diversity or spreading of the bacteria. At cool temperatures, more accurate data can be obtained or tested when arriving at the lab. Precautions such as having a rigid container to protect the samples during transport is also a must to prevent breakage and maintain temperatures, especially when using a courier to deliver the package to the lab. Keeping the culture sterile and tightly closed is important to allow no organisms or bacteria inside and maintain somewhat of a pure culture to avoid contamination. RT-PCR reverse transcriptase polymerase chain reaction assay can be used. IgG and IgM antibody enzyme linked immunosorbent or ELISA lab test can be done to confirm a viral infection.
How will you be able to confirm morphology of the virus, and what morphology could the virus possibly have? The morphology of the rift consists of an icosahedral 3-segmented genome of negative strand RNA in viral nucleocapsid protein. It is enveloped by a lipid bilayer containing 2 viral glycoproteins. One can confirm morphology of the Rift Valley fever virus with electron microscopy using the virus isolated in cell cultures. The cell cultures can be collected from the tissues of dead animals and aborted fetuses, particularly from the liver, spleen, and brain. The possible morphology of a virus depends on the shape of the capsid coat and could be a number of different shapes, such as icosahedral, helical, complex, or binary. The Rift Valley fever virus is a 3-segmented sphere, making it a 3-fold icosahedral. Can this virus be easily grown in cell cultures? Yes, the Rift Valley fever virus can easily be grown in numerous cell lines, including baby hamster kidney cells, monkey kidney or vero cells, chicken embryo reticulum, and primary cultures from cattle or sheep.
Which laboratory growth method is the best to use for this virus? The lab growth method that would work best for Rift Valley fever is an enzyme-linked immunosorbent assay, also known as ELISA. This is a serological test and allows the diagnosis of recent infections by confirming the presence of immunoglobulin M or IgM antibody to the virus. This method may be used in Rift Valley fever-free countries, as the ELISA test can be performed with inactivated antigen. Recently, the use of inactivated whole virus has been replaced by recombinant nucleocapsid N protein as the antigen. However, the ELISA test method may result in cross-reactions between other flaviviruses and Rift Valley fever. Other viruses and bacteria can also cause abortion in sheep and cattle. How will you confirm this is Rift Valley fever virus and not another virus or bacteria? In order to determine whether or not the virus is actually Rift Valley fever or another virus or bacteria, we would perform the ELISA test. If there is a positive test for IgM and IgG, the virus is Rift Valley fever. If there is a negative test for IgM and IgG, the virus is another virus or bacteria. For best results, at least 2 positive test results should be obtained from the same specimen using a combination of different methods of diagnostics, such as positive for viral DNA and antibodies, and positive for IgM and IgG. There should be 2 to 4 weeks between sample collection of paired serums showing raising titers between the pairs. In the ELISA test, IgM detection would suggest recent infection, whereas IgG detection would not differentiate between past and current infection.
Explain the specific eclipse phase of the replication of this virus. The eclipse phase is in the appearance of the virus within the cell. It is a period of time where the intra and extracellular virus cannot be detected. In the eclipse phase, the virus disassembles and the component parts are synthesized. The eclipse phase is simply when the virus has entered the cells but the virions have not been created and are not in its maturation phase. Rift Valley fever virus is a single-stranded RNA genome that has 3 segments: large segment, medium segment, and small segment. The L segment is the one that contains the RNA-dependent RNA polymerase L protein that is responsible for transcription as well as replication of the viral genome. The M segment is responsible for encoding glycoproteins that will allow the virus entry into the host cell. The M segment also contains NSM proteins that are responsible for maturation of the virus. The S segment is responsible for encoding the nucleocapsid and NSS segment. The dendritic cell-specific intercellular adhesion molecule 3 grabbing non-integrin is a receptor for the Rift Valley fever virus and binds directly to the attached oligosaccharides on virions. Viral entry will happen on lipid rafts through endocytosis occurring after the attachment. The L protein that comes from incoming virions will begin primary transcription. The viral polymerase will then cleave the capped host mRNA to prime the synthesis of viral mRNA, which is known as cap snatching. From here, viral proteins will accumulate and replicate, which enters the maturation phase associated with the NSM and NSS proteins. Is this virus prone to mutations, and what are the possible type of mutations that can occur in this virus? Since the virus was found approximately 120 to 130 years ago, it has developed genetic mutations that have created multiple strains found today. The RVFV genome is generally stable due to a lack of pressure needed to adapt because the virus is so successful in infecting its host. Most mutations found seem to come from substitution of amino acids in the proteins. These different mutations resulting in closely related strains could have effect on things such as infection, virulence, and transmission. This virus is not particularly prone to mutations, but does have a temperature-sensitive mutation. The most important mutation this virus undergoes is the MP12 mutation. When this genome is exposed to chemical mutagens such as F-fluorouracil, and it creates mutations in all 3 of its genome segments. The mutation in the M and L segments significantly lessen the effect of the virus, with the S segment contributing a small impact to lessen the effect of the virus. The mutation is temperature sensitive, meaning that it fails to replicate at 41 degrees Celsius. This means that viral replication is limited at these temperatures and it attenuates the virus effectively in the body. This vaccine has proven successful with minimal effects seen in ruminants and is licensed for use.
How does this virus enter the host, transmission of the virus, and how is this virus shed into the environment? Rift Valley fever is transmitted via a vector, the Aedes mosquito. This is known as transovarial transmission. It is also zoonosis and is transmitted to people who come into contact with blood, body fluids, or tissues of infected animals. This could be as a result of handling, slaughter, or veterinary procedures. Being an intracellular pathogen, which mechanisms can this virus use to evade the immune system of the host? The Rift Valley fever virus is able to escape the immune system via the evasion of neutralizing antibodies. Neutralizing antibodies defend a cell from an antigen or an infectious body by neutralizing any effect it has biologically. The virus contains an NSS protein, which is known as the virulence factor, that counteracts the antiviral effects of the type 1 interferon response. This protein also interacts with a repressor complex to inhibit the transcriptional activation of the IFN beta gene, which is involved in innate immune response. The NSS protein also has the ability to degrade protein kinase R, which protects the body against viral infection. With all these capabilities, the Rift Valley fever virus is able to evade the immune system and is thus considered a highly dangerous virus.